There was a question about the quizzes this week. So in laboratory, it'll be standard laboratory quizzes. There'll be a weekly quiz when you come into laboratory today on the laboratory we did last week. What did we do last week? Osmosis 2? Yeah, okay, so there'll be a weekly quiz on Osmosis 2 when you come back into laboratory today. And then at the end of today's laboratory, which is a photosynthesis laboratory, there'll be an exit quiz on that. And then Wednesday's laboratory will have its own exit quiz. Wednesday's lab is another photosynthesis lab. You get 2 photosynthesis labs. But there is no school on Friday. It's Cesar Chavez Day. And so there's no Friday quiz in here because of that. Sound good? All right. And let's see. So we were on this chapter on mitosis and cellular division. And let me forewarn you a bit. Today's lecture gets a little bit detailed in terms of the enzymes and procedures that cells use to carry out mitosis. So brace yourself for a little bit of molecular detail in this lecture. Kind of hate to club you over the head with a lot of detail on a Monday when you're probably still a little bit sleepy from the weekend, but unfortunately it's just what the lecture is today. Okay, well let's start off with a quick review. This lecture is all about how cells reproduce themselves, and how they do it is essentially just splitting in two. And I'll turn down the lights here in a moment, but I want to do some review on the blackboard here for a second. And so the cell that divides is called the mother cell. There's mother cell, and the cells it manufactures when it divides, when it splits in two, are called the daughter cells. And there are certain steps that the cell goes through when it divides. If here's the original mother cell, and there's the nucleus, the first step in the cell dividing is that the nucleus divides. They sometimes say that the nucleus splits in two, and the two nuclei go to opposite poles of the cell. One goes to the top of the cell, one goes to the bottom of the cell. And that step is called nuclear division. Division of the nucleus. And then, after the nucleus has divided, then and only then does the rest of the cell divide. So here's where you finally get the cytoplasm and the organelles divided into the two daughter cells like that. And that step is called cytokinesis. Like so. So it's always nuclear division first, and then the cytokinesis takes place. And there are 2 types of nuclear division, 2 styles of nuclear division, you might say. In one of them, called mitosis nuclear division, the nucleus divides in such a way so that both daughter cells get the same number of chromosomes that the mother cell had. So if this was a human cell, for instance, human cells have 46 chromosomes per cell, and that means the daughter cells would have 46 chromosomes per cell. Now, why does it say 2N up there? Well, because remember that in humans, the 46 chromosomes is actually 2 sets of 23. You have 2 chromosome number 1s and 2 chromosome number 2s and 2 chromosome number 3s, all the way down to 2 chromosome number 23s. 2 sets of 23 give you your 46. And so that just means that each of the daughter cells is also going to have 2 sets of our 23 chromosomes. That is mitosis nuclear division. Same number of chromosomes, 2 sets of chromosomes per cell, in both the mother cell and the daughter cells. Whereas in meiosis nuclear division, the daughter cells each get 1 set of chromosomes. So if this was meiosis, the mother cell has 2 sets of 23, but each daughter cell would only have 23 chromosomes, 1 set of 23.
Yes.
Yes.
So for mitosis, when mother cell like replicates the mother cell, like creates two daughter cells. And say those two daughter cells divide, do they become mother cells when they?
Yes, yeah, exactly. So when this mother cell divided, and now there are two daughter cells here, each of those can then divide and make its own two daughter cells. Then each of those can then divide and make its own two daughter cells. And so that's, I think that's mathematically, it's called exponential growth, where you keep doubling with every generation. And, you know, exponential growth is a very robust type of growth, and that's what allows one tiny little zygote, the fertilized egg cell, to grow to a newborn baby with about 4 trillion cells just in the space of 9 months. Okay, and we're going to be focusing for the most part, well actually completely, for the rest of this lecture on mitosis. So everything that follows from this point on is focusing on just mitosis. We are going to talk about meiosis, but that'll be in a couple weeks. So I'll give it as one of the pre-lab lectures. Okay, so the first step is in mitosis is nuclear division. Before nuclear division comes along, the cell has to prepare its chromosomes for nuclear division, doing something called DNA replication. Maybe I'll put another arrow here. Which is a same way of saying is that the chromosomes have to duplicate. All right, so this DNA replication step or chromosome duplication step takes place before the nucleus divides in cells that want to divide themselves. Before nucleus divides, the cell must replicate its DNA, and another way of saying the same things is that the chromosomes become duplicated chromosomes. Here's what it looks like. Here's an unduplicated chromosome, and you can tell it's an unduplicated chromosome because there's only one chromatid attached to the centromere. There's the centromere, and remember the chromatid is the piece of long double-stranded DNA, and the histone proteins is wrapped around. And when a chromosome becomes a duplicated chromosome, it now looks like this. There are now 2 identical chromatids attached to the original centromere. In other words, in a duplicated chromosome, the chromosome has photocopied its original chromatid, and so now it has 2 chromatids attached to it. And so notice that the chromosomes, duplicated chromosomes, have this sort of X-shaped look to them. Now, one of the human chromosomes, one of the 46 chromosomes, is called the X chromosome. Don't mix that up, that one chromosome, which is known as the X chromosome, with this. All 46 of your chromosomes will get this duplicated X shape when your cells are ready to divide. Here's a picture of some chromosomes from some cell's nucleus, and you can see they all have that X shape. So you would say these are all duplicated chromosomes. And if you saw this under a microscope, you'd know that this cell was getting ready to divide, because this always happens. This only happens when the cell is going to be dividing. All right, and there's some terminology related to the duplicated chromosome. The 2 chromatids that are attached to the centromere are called sister chromatids. And they are utterly and completely exact copies of each other. If there was, let's say, a gene here for eye color, and it happened to be the blue allele of the eye color gene, you would find the blue allele of the eye color gene here. And if there was a gene here for, I don't know, kind of, let's say, pimples or something like that, then you would find that same gene for pimples over there. Each sister chromatid is a complete and utter clone of the other sister chromatid. But an important point here is it's still considered one chromosome. Despite the fact you might be tempted to say, well, that's 2 chromosomes. Yeah, there's 2 chromatids there, but because there's one centromere, it counts as one chromosome. And we're going to be doing some work with going through the various steps of mitosis in the next few lectures, and I'll be asking you to keep track of the number of chromosomes in the cell. And so what you'd always do is just focus your attention on the centromeres. However many centromeres there are, that's how many chromosomes there are, because by definition, one centromere equals one chromosome, no matter how many chromatids are attached to that centromere. Alrighty, well, let's see. Let's go back in time for a second to the chromosome before it duplicated. Oh, actually, there's a nice photograph of one, kind of neat. Looks almost fuzzy. That's the DNA all folded up on the histones that gives it that fuzzy look. There we go. So here's this chromosome that's going to be duplicating for us here in a minute. What I want to do here is begin to talk about the molecular detail of how the chromosome replicates the DNA. In other words, how it goes from an unduplicated to a duplicated chromosome. And let's see, to introduce the details of how the duplication occurs, I want to give a very simplified version of the chromosome. So here it is like this. Maybe I'll draw it even more simply on the board like this. So here's one piece of DNA in the chromosome, there's the other piece of DNA in the chromosome. And remember that it's double-stranded DNA. And you might remember one of the rules of double-stranded DNA is they always go in opposite directions, just like the two lanes on the highway go in opposite directions. The 5' end of a piece of DNA is considered its beginning end, and the 3' end of a piece of DNA is considered its ending end. And so this top strand goes from that side, from left to right in this diagram. The bottom strand has to go in the opposite direction because that's just the way double-stranded DNA works. Like that. Okay, so that's how we'll start off the chromosome. Now, this chromosome here is only about, looks like 10 base pairs long. If you count the base pairs, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And that's just absurdly small because in real life, chromosomes can be hundreds of millions of base pairs long. But just to illustrate the process, I want to simplify things a little bit. Okay, and again, just to make the diagram a little more clear, I'm going to get rid of the centromere. It's actually still there in real life, but let's just make it invisible for a moment. Okay, so when this cell wants to duplicate its chromosomes, the first thing that it does is the double-stranded DNA strands separate from each other into two single DNA strands. So let's separate those guys. There, 5' down there like that. Now you may wonder, well, how does the cell do that? Well, there are enzymes that do it. There's an enzyme called helicase. You don't have to know the name of that one, but there's an enzyme called helicase which unwinds the DNA and splits the double-stranded DNA into two single strands, like so. Now, what's going to happen next is there's going to be an enzyme that comes along and latches onto the single-stranded DNA to make complementary strands to it. In other words, an enzyme, which is actually called DNA polymerase III, is going to latch onto those strands and build a complementary strand to each one. And let's take a moment just to realize that that's what we're looking for here. We're looking to go from one piece of double-stranded DNA, one chromatid, into two pieces of double-stranded DNA, two chromatids, and this would do it. If we can make a complementary strand to each of these single strands, we do end up with two sister chromatids, two identical pieces of double-stranded DNA. And anyway, DNA polymerase III is the enzyme that's going to be the main enzyme in duplicating the DNA. Okay, and here comes DNA polymerase 3. There she is, all right. Who remembers where we saw RNA polymerase? What process was the enzyme RNA polymerase involved in? It was... Nobody remembers RNA polymerase? To give you a hint, it latched onto this strand and made an RNA complementary strand to it. Nobody remembers RNA polymerase? It was in transcription, where cells are expressing a gene. This RNA polymerase enzyme latched onto this strand and made a complementary RNA strand, which was called the what? It's an RNA strand that left the nucleus, went to the ribosomes. What do we call that RNA? Messenger RNA. Yeah, I think I heard it. Anyway, so RNA polymerase latches onto the strand and makes a complementary RNA to it, which is the messenger RNA that goes to the ribosome. But this DNA polymerase 3 is not going to make an RNA strand, it's going to make a complementary DNA strand. This DNA polymerase 3 enzyme, the strand that it binds onto is always called the template strand. And this enzyme has some rules it has